Hi, it's Dana from We'll Talk with an ingredient inspector investigation on cholesterol. This is part 2 of cholesterol, where we look primarily at lipoproteins. 2018. What are lipoproteins? The definition: lipos is the Greek word for fat. A lipoprotein is fat and protein. Before entering our bloodstream, cholesterol is paired with fats and proteins and other items to form lipoproteins. Cholesterol is contained within lipoproteins. Their functions are primarily to deliver and pick up various substances to and from cells. Lipoproteins are how cholesterol and fat and certain other items are transported through our bodies in partnership or as a component of lipoproteins. The primary substances transported by lipoproteins include cholesterol, fat as triglycerides, specific proteins, and fat-soluble antioxidant vitamins such as A, D, E, K, and CoQ10. What are the 5 key types of lipoproteins? There are 5 key types: chylomicrons, very low-density lipoproteins, intermediate-density lipoproteins, low-density lipoproteins, and high-density lipoproteins. We'll also take a look at LPA at the end of the video. How are lipoproteins designed? Lipoprotein particles consist of an outer layer or a membrane and an inside core. So there's the outer membrane and an inside core. There's also a layer between these two called the hydrophobic interfacial layer. Hydro means water, and phobic is from phobia or fear of. So this interfacial layer is kind of like a revolving door between the inside and the outside. And it's hydrophobic, keeping the inside waterproof and safe. We're going to focus really on the inside core and the outside membrane. The circle is an easy shape for information purposes. It's not actually completely circular like we're picturing here. The actual shape is what's described as a disc-shaped particle. So it's a little more like if you took a round circle and sort of smashed it a bit, it wouldn't be perfectly circular. It would be what they call disc-shaped. What is the general composition of the outer membranes of lipoproteins? They vary, so we're just talking here about 3 of the primary ingredients in terms of the general composition. They're made primarily of phospholipids, apolipoproteins, and free cholesterol. So phospholipids, it's phosphate plus glycerol plus 2 fatty acids, which are lipids, are phospholipids. The glycerol acts as an ester, helping to bind the phosphate and the lipids together. The fatty acids consist of long chains of carbon and hydrogen with 2 oxygen atoms at one end. They help to make the lipoprotein particle waterproof. Apolipoproteins, or protein, some only have one, some have more than one on the outside. They are proteins that form certain biochemical molecules like enzymes. So apoprotein plus lipid is the apolipoprotein. And the lipo part comes from the lipid, which means fat. So the apoproteins are the directors. They facilitate the lipoprotein's delivery process to the cells. They oversee what is going on with the lipoproteins as they move through our bodies. They also contribute to the structural integrity and the solubility of the membrane. And free cholesterol. A small amount of it is woven into the membrane between the phospholipids to keep them from joining and hardening or crystallizing. So first we'll take a look at chylomicrons. So there's the lipoprotein. What is the composition specifically of chylomicrons? In the inside core, we see triglycerides and cholesterol esters. Triglycerides is in a larger box and font size to communicate that there are more triglycerides than cholesterol esters in the core of chylomicrons, and we'll keep that kind of visual consistent throughout the video. So when the box is bigger, it means that there are more of them. In the outer membrane, We see the phospholipids and the apolipoprotein, in this case apolipoprotein B, specifically B48, and the free cholesterol. Those are the three things that we just looked at before. We also see in the outer membrane apolipoprotein A1. This A1 and also other apolipoproteins are in the outer membrane of a chylomicron. It also contains fat-soluble antioxidant vitamins. And we'll take a look at antioxidant because that comes up a lot just in our world, but also here specifically, it plays a very important role. So the triglycerides and the cholesterol esters. What are antioxidants? Oxidation or antioxidants. Antioxidants combat or fight harmful oxidation. Oxidation is the state of being oxidized or exposed to oxygen. Oxidative damage is when things are exposed to and damaged by oxygen. They go bad or turn rancid. For example, if you cut an apple open and leave it out, what you see as it turns brown is the effect of oxidation. In the outside layer or membrane of lipoproteins are numerous fat-soluble antioxidant vitamins. Fat-soluble means fat must be present for the vitamins to be dissolved. The lipoprotein delivers these essential antioxidant vitamins to cells, but these vitamins also serve as protection for the lipoprotein itself as it moves through our body. So it's delivering these to cells, but it's also protecting the lipoprotein itself. Fat-soluble antioxidant vitamins are like tiny fire extinguishers that neutralize harmful oxidation within the lipoprotein particle. In our bloodstream, antioxidant vitamins turn potentially harmful molecules into harmless molecules. These helpful passengers serve to keep the lipoprotein itself healthy and functioning efficiently as a transport vehicle. So by having these antioxidants on board, contained within it, the lipoprotein basically has its own medical team keeping it in shape as it moves through our bloodstream, delivering its various essential items. What are triglycerides? Triglycerides are 3 fatty acids or lipids plus glycerol. The tri in triglycerides stands for 3, like think of a tricycle with 3 wheels. Triglycerides are fat, so when you see triglycerides, just think fat. Glycerol acts to bind the fatty acids together. There's a visual of it. So there are 3 fatty acids. The glycerol is what holds the 3 of them together. It's sometimes referred to as the glycerol backbone. And then we see these red points. These are the esterification sites where ester bonds are formed, where the fatty acid can connect to other items. What are the differences between free cholesterol and cholesterol esters? There are 2 forms of cholesterol in our bodies, free cholesterol and cholesterol esters. Free cholesterol is an active form. It can be used in this form, whereas cholesterol esters are a storage form. It stays in this form as it moves around, being stored until it needs to be used, and then it is transformed into free cholesterol. So the free cholesterol active form, readily available, able to be absorbed, and the cholesterol esters are not readily available. Free cholesterol is also referred to as unesterified cholesterol, whereas cholesterol esters are esterified. Free cholesterol molecules are amphipathic. That means they contain both polar, hydrophilic, and nonpolar, hydrophobic properties. Hydrophilic means attracting water, and hydrophobic, which we talked about a few minutes ago, is about repelling water. Cholesterol esters are nonpolar only. They are hydrophobic molecules that repel water. They are insoluble in the blood. Insoluble means it cannot be dissolved. So free cholesterol is single and a cholesterol ester is attached. What are chylomicrons? Chylomicrons are the largest type of lipoprotein. They are created in our guts in the cells of intestines, also known as enterocytes. Chylo is a derivative of chyle, which means juice, and micron, which means small particle. Chylomicron's primary role is to carry fats from food we eat through the bloodstream, to deliver them to cells in our muscles and to our fat storage cells for future use. Fats from food we eat are referred to as dietary lipids or dietary fat. The fatty core of chylomicrons consists mostly of triglycerides, approximately 85%. Chylomicrons are the lowest density lipoprotein because they have a low ratio of protein to lipid. The triglycerides of chylomicrons are created from the digestion of dietary fat. Chylomicrons are mostly triglycerides because this is how fat fuel is delivered to muscles and fat cells, which are also known as adipocytes. Chylomicrons are released into our bodies via the small intestine into the lymphatic system. Chylomicrons go from our gut through our lymph nodes, then into our heart. The body is designed to deliver cholesterol and fat-soluble vitamins to our heart first. Chylomicrons also contain small amounts of cholesterol esters and free cholesterol. Similar to the triglycerides in chylomicrons, these cholesterol forms are derived from food consumed. These are considered exogenous cholesterol, or cholesterol derived from outside the body. Ultimately, chylomicrons are responsible for moving cholesterol and fat from the gut to the liver. The chylomicrons still have cholesterol and some triglycerides when they return to the liver. What are the functions of chylomicrons? Once in the lymphatic system, chylomicrons are helped along with additional specific apoproteins from existing Vldls, which are very low density lipoproteins, and HDLs, which are high density lipoproteins. The addition of these apoproteins takes the chylomicrons to maturity. In their outer membrane, these mature chylomicrons now carry specific proteins that will help create HD particles, sometimes referred to as baby or nascent HDL. Chylomicrons move through the bloodstream to deliver fats and cholesterol to specific tissues. When chylomicrons encounter hungry cells in muscles and adipose fat tissues, the triglycerides in the chylomicrons are utilized to feed the cells the fuel they need. The triglyceride is broken down by lipoprotein lipase, also known as LPL. Lipoprotein lipase is an enzyme that breaks the triglycerides into its components. During this fuel transfer, phospholipids and fat-soluble antioxidant vitamins in the chylomicron's outer membranes may also be absorbed into the cell. Once their work is done delivering essential fuel and nutrients, chylomicrons will give up some of their phospholipids to HDLs. The chylomicrons are broken down as their components or parts are consumed by needy cells, and only remnants or pieces are left, which are referred to as chylomicron remnants. Chylomicron remnants have little or no triglycerides left. They are richer in cholesterol because the triglycerides were depleted. The cholesterol-containing chylomicron remnants will find their way to the liver. This is how chylomicrons move dietary cholesterol and fats from the gut to the liver. So let's take a look at VLDL, IDL, and LDL. What are VLDL, IDL, and LDL? Very low density lipoprotein, intermediate density lipoprotein, and low density lipoprotein. So 3 types of low density lipoproteins, 3 variations in density. The ratio of lipids to protein influences the density of these lipoproteins, but this trio is interrelated. VLDLs become IDLs, which become LDLs. So very low density lipoproteins are synthesized by the liver in a process called lipogenesis, or the beginning of fat formation. These newly constructed VLDLs are sent from the liver to the bloodstream. In the bloodstream, as they do their job moving about, they slowly become intermediate density lipoproteins, and ultimately they become low density lipoproteins. So LDLs are technically formed in the circulation from VLDLs. What are the differences in composition between the 3? So there's the three. The lipoprotein begins as a VLDL, but its density shifts as the particle gives up triglycerides to hungry cells. So VLDLs become IDLs, which become LDLs. This is referred to as the delipidation cascade, also sometimes referred to as the lipolytic conversion. So we see the composition of the three here, and we see how it changes from one to the next. Start out with a lot of triglycerides and end up with a little. Start out with a little bit of cholesterol esters and end up with a lot. So VLDLs are very low density because they contain 5 times more triglycerides than cholesterol esters, and triglycerides are not as dense as cholesterol esters. And on the other end, LDLs are low density because they contain more cholesterol esters than triglycerides. What is a Vldl remnant? That's another term you might come across. Basically, as the VLDL is moving through the bloodstream and the triglycerides are depleted and it picks up more cholesterol and its composition begins to shift, there is something called a Vldl remnant that is actually very similar in terms of composition to an IDL. So sometimes they really are referred to as the same thing. So in the bloodstream, we have VLDL remnants, LDLs, and IDLs moving about. The LDL receptors are most prominent in the liver. Those receptors that are going to identify these items and pick them up out of the bloodstream. So the liver picks up LDLs, but it also picks up the VLDL remnants and the IDLs. And it's because all three of these low-density lipoproteins have the same apoprotein B, specifically B100 component, that allows the liver's receptors to recognize them. So these all enter into the liver. When picked up by the liver, this trio of lipoprotein types has only a small amount of triglycerides and phospholipids left. The triglycerides and phospholipids were either traded or consumed. All three of these lipoprotein types are rich in cholesterol esters when they return to the liver, because they picked up additional cholesterol while in the bloodstream. This trio of LDLs return the majority of cholesterol in the bloodstream to the liver, and this is referred to as indirect reverse cholesterol transport. For a long time, it was believed that HDL solely did the job of bringing cholesterol back to the liver, but we now know that these LDLs also return cholesterol to the liver. So once in the liver, some of the cholesterol from these items is kept by the liver to incorporate into its cell's membranes. Portions of the three lipoproteins are metabolized into bile acids. Portions of the three lipoproteins are also used to create new, very low-density lipoproteins. The remainder of the lipoproteins, as LDLs, are sent from the liver directly to the intestines. Some LDLs are also sent to the intestines indirectly via the biliary route, through the bile. Only free cholesterol can be sent from the liver in this way. In the intestines, excess LDLs are eliminated. This is how the body balances the overall cholesterol levels, by getting rid of excess. Remaining LDLs enter into the bloodstream. What is LDL receptor efficiency? The LDL receptors in the liver are especially important since the liver is where these lipoproteins will finally leave the bloodstream. Ideally, we want rapid clearance of LDLs from the bloodstream. The best scenario is when production of LDLs and clearance of LDLs is in balance, so there's no accumulation of them. The functioning of the LDL receptors in the cells is of crucial importance. How efficient are the LDL receptors at taking up the lipoproteins? How well do the receptors metabolize the lipoproteins? The LDL receptor itself can become impaired or have functioning reduced by oxygen if it has undergone oxidation. Various other factors may also contribute to an altered or changed structure to lipoproteins. These changes influence how easily the particle is recognized by the receptor and how easily it is assimilated by the receptor. Some lipoprotein particles fit into the LDL receptors better. Some are more efficiently internalized into the cell. As the lipoproteins move through the bloodstream performing their functions, the diameter and shape of the particles change slightly. Tiny structural modifications based on lipid and protein interactions within the particle alter its size and shape. The ApoB100 conformation may also be slightly altered based on how the particle diameter and core lipids composition changes, which oils and how much. LDL particles may have slightly different charges on their surface based on their altered shape, positive versus negative. So a higher negative charge on the particle's surface reduces the affinity by receptors. So then there's a reduced LDL clearance rate, which leads to the LDLs remaining in the bloodstream longer, which leads to enhanced risks. So this is plasma residence time. How long do these low-density lipoproteins stay in the plasma? And as you can see, there are differing times here. The LDL has the highest amount of time with 1.5 to 2.5 days in the plasma. So we're going to focus on that. The outer membrane of LDLs contain polyunsaturated fats, which are most prone to oxidative damage. The longer LDLs are in the blood and not being taken up by cells, the more likely that protective antioxidants are depleted, the more likely oxidation will occur. Insufficient antioxidants lead to insufficient defenses, which lead to oxidation. Once the fat or triglyceride component inside an LDL particle is oxidized, it attracts white blood cells, which initiate inflammation for protection. Also, the oxidized LDLs are not taken up by cells that are searching for fat. Then the damaging free radicals attack and oxidize the LDL's protein component. If the protein component of any lipoprotein is oxidized, the LDL receptor will not recognize the lipoprotein and will ignore it. This is called impaired recognition. Oxidized LDLs cannot be recycled by our liver because our liver receptors that search for specific LDL particles don't recognize those LDLs that have been damaged by oxidation. These damaged LDLs stay in the bloodstream, and we know that that is not a good thing. How does the composition of lipoproteins affect their functioning? The triglycerides in lipoprotein particles are comprised of fatty acids and glycerol. But which fatty acids the triglycerides are made of is important because this alters or changes the strength or structural integrity of the particle. Fat, we know, becomes oxidized when it is exposed to oxygen. So depending on the source of fats in the lipoprotein, that is the degree of the potential for oxidation. We know that the lipoproteins contain antioxidants to keep the fats from turning bad or oxidizing. If you recall this visual that we looked at, that the antioxidant vitamins are being delivered to the cells, but they're also protecting the lipoprotein itself. So the lipoproteins can be destabilized if the source of fats is primarily vegetable oils, like corn, sunflower, safflower, canola, cottonseed, and soy. Vegetable oils are not ideal building blocks for lipoproteins because they oxidize quickly. Often when the lipoproteins are made with these oils, there are not enough antioxidants within the lipoproteins to neutralize the oxidation. Some of the fat in the lipoproteins goes bad. Vegetable oil will oxidize sitting on a counter, just like that apple, and this can also happen inside our bodies, but we cannot see it. Cholesterol and fat do not clog our arteries. Fatty foods are digested and packed into particles that can carry fat in our bloodstream without clogging them up, but only as long as those particles themselves are properly built. When our diets are full of corn and soy oil, the lipoprotein particles do not contain enough antioxidants. Without antioxidants, lipoproteins are easily destabilized. The fat they were shuttling through the bloodstream is suddenly unprotected and cannot stay suspended in the blood, so it will splat onto the interior lining of the artery just like a paintball. This is a quote from Dr. Catherine Shanahan, the author of Deep Nutrition and Food Rules. What is ApoB? Apolipoprotein B, or ApoB, performs many essential beneficial functions, but ApoB has the unfortunate ability to deliver LDLs into the walls of arteries or blood vessels. The endothelium tissue, which lines the inside of our arteries, is only 1 cell thick. So there's this very thin lining along the inside of our arteries. Blood vessel walls have small spaces that ApoB containing LDLs can move through the endothelium to reach, spaces like behind or beneath the arterial wall, effectively parking themselves there, so they can get behind and in between that space. The endothelial cells create a border that protects the arterial wall, but an ApoB containing LDL can penetrate that border when the endothelial cells are injured or inflamed. This is referred to as retention. The LDL particles stay there. Subendothelial retention. Sub means under or beneath, like a submarine. Once in subendothelial space, oxidation of the LDL particle begins to occur, and we know that this is a bad thing. Oxidation leads to an immune response by our bodies, which leads to inflammation. Our bodies can deal with the inflammation in the beginning when we are young. The threat is more easily eliminated at this time. Arterial wall macrophages, which are phagocytic cells which can absorb bacteria and other potentially harmful items, can actually move the cholesterol from these damaged LDL particles to HDL particles in the bloodstream. However, it can become increasingly difficult for our bodies to keep up. Chronic or all-the-time inflammation leads to atherosclerosis, which leads to heart disease. What are Pattern A versus Pattern B LDLs? There are 2 types of LDL particles, Pattern A and Pattern B. Pattern A and Pattern B have different densities and diameters. Pattern A particles are large and buoyant. Pattern B particles are small and dense. Although Pattern A and Pattern B are differentiated by size and density, the size does not determine the amount of cholesterol in an LDL particle. A large Pattern A and a small Pattern B particle could have the same amount of cholesterol. So this is not about the amount of cholesterol, it's the way in which the LDL is packaging it, either large and buoyant or small and dense. What are the defining characteristics of Pattern A versus Pattern B LDLs? Pattern A particles are big and buoyant. They're often referred to as large and fluffy. It's the ideal state for LDLs. Pattern B particles contain less fat and so are small and dense. They tend to sink in the bloodstream due to their density. Because the protein element is exposed, Pattern B LDLs are more vulnerable to oxidation than Pattern A. The oxidation damages the LDL particles, so the liver does not recognize them. And as a result, we know what this means. Pattern B LDLs remain in the bloodstream longer than Pattern A. The smaller size of Pattern B also allows them to move into smaller spaces, to penetrate arterial walls, and this of course poses a greater risk. Pattern A, each particle is surrounded by fat, and so these larger particles are slower to oxidize than Pattern B. Pattern B is considered more atherogenic. It promotes the formation of fatty plaque in the arteries, which is referred to as atherosclerosis. What are the differences in Pattern A versus Pattern B LDLs with regard to total LDL cholesterol? So if total LDL cholesterol content is the same for Pattern A and Pattern B, like the same size of bread loaf in this analogy here, there will be more pattern B particles by definition because they're smaller and dense. So we see the bread sliced up under pattern B to represent the idea that even if it's the same amount, there will be more of these particles when they're pattern B. Impaired glucose function can increase pattern B LDL particles. Small LDL particles are caused by too many simple carbohydrates and excess sugar, which leads to high triglycerides. The LDLs become unable to deliver to cells because there are sugars stuck to them, hindering their function. So these LDLs are not picked up and recycled by the liver, and they stay in the bloodstream as small, dense particles. These are the types of foods that are simple carbohydrates that can lead to high triglycerides. HDL. What are HDLs? HDL stands for high density lipoproteins. HDLs are the smallest lipoproteins and have the highest ratio of proteins to lipids, therefore they have the highest density. HDL particles are created or synthesized primarily in the liver but also in the small intestines. HDL particles are also created in the bloodstream itself from the nascent baby HDLs. In their core, HDL particles begin as approximately 90% cholesterol esters and 10% triglycerides. This is one of the ways the liver gets rid of any excess cholesterol. The larger an HDL particle is, the more cholesterol it carries. HDLs also contain various apoproteins in their outer membrane, including 1 to 5 copies of apoprotein A1. Apoprotein A1 activates the enzyme lecithin cholesterol acyl transferase, which is responsible for converting free cholesterol to cholesterol esters for storage. What are PLTP and CTP? These are two specialized glycoproteins that are released into the circulation to facilitate the HDL transfer activity. So our bodies release these things into the plasma so that when the HDL needs to do its job, these two glycoproteins are there to help facilitate it. Plasma phospholipid transfer protein, PLTP, oversees the phospholipid transfer activity of HDL. It helps to regulate the metabolism of the HDL particle, keeping it healthy and in balance. So this is referred to as PLTP-mediated phospholipid transfer. CTP is cholesterol ester transfer protein. It oversees cholesterol transfer activity. CTP-mediated cholesterol transfer. It also catalyzes the transfer of neutral core lipids between HDL and other lipoproteins. This is something that PLTP cannot do. CTP is apoprotein D. What are the functions of HDLs? HDLs perform numerous functions, some of which are related to cholesterol and lipid transport. Lipidation and delipidation. HDLs spend about 5 days in the bloodstream undergoing lipidation, which is acquiring cholesterol, and delipidation, delivering cholesterol. It's transferred from cells and other lipoproteins. HDLs deliver cholesterol esters to and pick up free cholesterol from fat storage cells, which are known as adipocytes. HDL particles are in a state of constant flux. The particles shift in size and composition. HDLs deliver essential cholesterol esters to adrenals, testes, and ovaries to help the production of steroid hormones. Tissues in these areas are called steroidogenic tissues. With the assistance of CTP, cholesterol is taken up by these organs by sterol influx transporters. If necessary, cholesterol can also be exported by these organs to HDLs via sterol efflux transporters. So cholesterol influx and efflux taking place. Also in the functions of HDLs, homotypic and heterotypic exchange. HDLs trade lipids with one another. This is referred to as homotypic exchange. HDLs also swap cholesterol esters with VLDLs and LDLs for triglycerides. This is referred to as heterotypic exchange. CTP is also used in this exchange between the lipoproteins. HDLs become more triglyceride-rich while in the bloodstream, while VLDLs and LDLs become more cholesterol-rich. HDLs trade proteins to LDLs for cholesterol and fat-soluble antioxidant vitamins, especially vitamins D and E. HDLs deliver vitamin E to endothelial cells that line the walls of blood vessels. What are direct and indirect reverse cholesterol transport? HDLs can move the cholesterol component of parked LDLs from subendothelial space beneath arterial walls to the liver for elimination. The HDLs take up cholesterol from lipid-laden macrophages, which are foam cells, in the arteries via ATP-binding cassette transporter G1, APZ G1. This is referred to as HDL-mediated cholesterol efflux. The A1 apoprotein component of HDLs also take up cholesterol from the foam cells via ATP-binding cassette transporter A1, ABCA1, ApoA1-mediated cholesterol efflux. The cholesterol is esterified by the LCAT, and these cholesterol esters are selectively picked up by the liver via scavenger receptor class B type 1, SRB1. This is direct reverse cholesterol transport. Indirect reverse cholesterol transport is the HDLs transferring cholesterol to ApoB particles, 90% of which are LDLs, in exchange for triglycerides. These ApoB particles then return the cholesterol to the liver, so that's the indirect way. HDLs deliver cholesterol to the liver, but can also deliver some of the cholesterol to the intestines. This is known as transintestinal cholesterol efflux, or TICE. So those are the lipoprotein types, and we said we would also take a quick look at LPA. What is lipoprotein A? Lipoprotein A is a low-density lipoprotein and is abbreviated as LPA. LPA is an LDL-like particle with apoprotein B100, but it also has apoprotein A. So it has the B, but it also has A. LPA is a subset of LDL. Only some LDL particles contain LPA. Lipoprotein A has been identified as a genetic risk factor in coronary heart disease and stroke. Genetic means it's something inherited or given to us by our parents. High LPA can be part of our genetic makeup. LPA helpfully carries harmful oxidized phospholipids for the other lipoproteins, fats that have gone bad from exposure to oxygen. Oxidized LDLs transfer oxidized phospholipids from their membranes to the LPA particles, and LPA may also pick up oxidized phospholipids from other cells, like endothelial cells that line the walls of the blood vessels. LPA particles are like garbage cans in our bloodstreams. They serve an essential purpose, but they are not so nice. Measuring LPA is used as a marker for harmful oxidation of the LDL membrane. LPA may have enhanced penetration of endothelium. LPA is stickier than LDLs, so it has a greater adhesion ability, and LPA may have greater retention in subendothelial space. When you find them, LDL, stuck inside artery walls, you're not actually looking at LDL, you're looking at LPA. That's a quote from Dr. Malcolm Kendrick. Thank you to the sources for this video. So that's the ingredient inspector investigation into cholesterol. So you can make excellent decisions about what is best for you. The ingredient inspector on Twitter and YouTube. ingredientinspector.org. Share with the ones you love.